Right off the bat, I will say that I think Dr. Dan did an excellent job debating Kent Hovind. One really effective thing Dan did was to have one question in mind heading into the debate, and to keep asking Kent that question. While Kent is obviously an easy defeat, that doesn't mean that every debate goes the same way. When you debate Hovind, you will never convince him of the truth about science, or get him to admit that any of your points are correct. Kent and his fans have already decided that he's the winner before round one even starts. So the goal of the debater personally is they should keep their cool, be prepared, and relentlessly ask Kent their question. Try to control the flow of the debate and avoid letting Kent drag you around and gish galloping. The goal when it comes to handling Hovind is to publicly expose him and to show his dishonesty by answering his challenges and trying to corner him and get him out of his comfort zone by forcing him to go off script. The mistake that people make when debating Hovind is that they will ask him a question, but then instead of answering, he just gish gallops and changes the topic, and by the time it's their turn to talk, they've forgotten about their question. Or if they ask him a question more directly, Kent just dodges it. Kent does not listen to his debate opponents, and this has never been more clear than during this debate. The debate followed a format of Dan talks and brings up great points, and then he asks Kent his main question, and then during Kent's turn, Kent completely ignores everything Dan said and just reads from his script. Either that, or just repeat what Dan said and just say, "Oh, it's wrong," followed by saying, "They are teaching that animals have common ancestry." Seriously, that was so annoying. Like, instead of answering something that Dan said, he would just say, "Well, they are teaching that mosquitoes and bacteria have a common ancestor," or "Do you believe that animals have common ancestry?" It's like, yes, you already know the answer to this. When he did reference Dan's arguments, he would say, "We've never seen a single-celled animal go to multicellular," despite Dan giving examples again and again of algae. Kent also said that there's never been a mutation to allow someone to hold their breath for 3 minutes, and then Dan gave 2 examples. It's too bad the garbage Q&A section started after these examples, so Kent didn't respond to them. Of course, Standing for Truth didn't tell Kent to respond either, he just started the Q&A. Of course, I'm sure if Kent did respond, he would have just pulled something out of his ass and said something that was completely irrelevant to what Dan said, so I guess it doesn't really matter. And here are some highlights.
Here is the feathers in the bird on the bottom left there, and then the scales converted to feathers in the crocodiles on the right. This is the HIV, new biochemical function, on the right, the ancestral one on the left in that figure. This is the new type of green algae that has evolved from the amoeboid rhizarian. This is, well, you can't see it because there's that headline in the way, but that's one of the many lizards that lays eggs. And where I left off right there is that not only does this involve changes to the way the eggs develop, but in a few species of these lizards, they actually develop a placenta. So we have a completely independent evolution of the placenta in a group of reptiles facilitating live birth. And what I was saying right when Standing let me know there was a problem there was that we recently witnessed a lizard lay 3 eggs and then give live birth to the 4th member of that litter. So there are populations that are actually undergoing this change from egg laying to live birth right now. This is not some hypothetical thing. We're watching this evolution happen in real time. So how about some photosynthetic animals? Photosynthetic animals seem like something that would constitute a change in kind. If I asked a 5-year-old if that lizard that could do photosynthesis was different from a lizard that couldn't, I think the answer would be yes. So we see this not just in a few walking animals, we see this all over the place, in sponges, in cnidarians, in mollusks. And the cool thing here is that it's not just a matter of stealing the photosynthetic structures from your food, because you actually find the genes for photosynthesis in the animal genomes. And as you can see in these developing salamander embryos right here, it's actually passed vertically. So those already are green. They're expressing photosynthetic proteins and have photosynthetic structures, even though they're still developing in the egg. And one more example, what about multicellularity? a completely new instance of multicellularity. Now, this is directly observed in the lab. There's a species called Chlamydomonas that's usually unicellular. But if you introduce a predator into that population, they'll start to behave like colonies. And in at least one instance, we've evolved true multicellularity because throughout their entire life cycle, there's no unicellular stage. And you can see that on the part D right here. Part A on the left represents their ancestral state. Part D on the right represents their derived multicellular state. That requires a whole bunch of new traits, not just a couple of new traits, but you need traits for cellular interactions. You need traits for a membrane, an extracellular membrane to hold all that stuff together. And they actually reproduce via multicellular structures called propagules. And that was a completely new thing for this type of previously unicellular green algae. So that's a whole lot of examples of things that, I think pretty reasonably we could call some of that stuff macroevolution. Now, is this all imagination? No, it's absolutely not imaginary. These are things that we have directly observed. There's no hypotheticals looking at fossils or anything here. So I don't want to hear anything about inferred evolutionary assumptions, none of that stuff. This is stuff we've directly observed. So let's turn it around. Let's look at Kent's model here, because in science, throwing stones isn't good enough. You need to have a better explanation. Does Kent's model do a better job explaining extant biodiversity? No, Kent can't even explain Kent's model in a consistent way, and we're going to see why that is. Faster speciation and diversification in the creation model than you do in the evolution model.
You've got just over a minute, Dan.
Thank you. So, for example, in the approximately 300 years from the flood to Abraham, you need to get over 150 species of cats, all the extant cats plus all the extinct cats. And that's not just a problem for cats. These are examples of Egyptian hieroglyphics right here that date from the Middle Kingdom. So you've got things like elephants, giraffes, you've got baboons and monkeys, you've got different things, you've got hares, you've got all kinds of different birds, you've got vultures, you've got falcons, you've got owls, you've got, I don't even remember what they are. There's storks, there's flamingos, there's sparrow and swallow. You have extant biodiversity within a few hundred years after the flood, and the creation model has no way to account for that. On the other side of things, I've given you examples of observed massive evolutionary changes, and if anyone wants, I can describe the mechanism for every single one of them.
Awesome. Just on time with 3 seconds to spare. So I appreciate that, Dr. Dan. I'm going to reset the clock, 6 minutes. Dr. Hovind, whenever you're ready, I'll start the timer.
Well, yeah, he mentioned things are observing right now as evolution, but what he gave is simply variation within the same kind. Nothing's changing into a new kind. Some reptiles lay eggs, some lay eggs, and some hatch the eggs internally, and the baby comes out alive. There's ovoviviparous and viviparous. I'm sure you're aware of that, studying biology. So I don't know that there's anything new has happened. They're certainly not developing the ability to split in half or develop without a male fertilizing the egg.
That's where you're wrong, Kent. Now, salamanders aren't reptiles, they're amphibians, but Kent Hovind probably wouldn't know the difference anyway. The female Jefferson salamander is able to pick up sperm deposits left behind by other salamander species and use it for genetic cloning of itself. Not fertilization, it makes a genetic clone that's an exact copy. These sperm deposits that are left behind on leaves and twigs are from other species of salamander. The Jefferson salamander can incorporate the DNA from these other species into its own genome when it makes a clone. This is important to the survival of the Jefferson salamander because if a species is genetically identical, they can be very vulnerable to diseases. This is how the Irish potato famine happened.
I mean, so what you've given us are examples of variations within, they're still the same kind, still a lizard. That's not evolution. Somehow you need to get your lizard to come from a rock over billions of years or come from an amoeba. Well, I'll let you start off with a living form like an amoeba. To go from single cell to multi-cell. You don't have any examples of that.
The lizards with the live births, some of them they basically have shellless eggs, but several species do have actual placentas. That's not up for debate. They actually have evolved placentas separately from mammals. It's a brand new placenta, and that requires a bunch of new genes and protein-protein interactions. And again, this is something we're watching happen in real time. So it's not like that's you can't really deny what's cannot you can watch what happen. Magnitude of change, what type of characteristic cannot evolve, and what specifically is the mechanism that prohibits it. That's where I want to leave that.
You guys believe the elephant and the dog all came from a single cell creature like an amoeba. We don't ever see single cell creatures produce anything other than single cell babies. Never.
De novo origins of multicellularity in response to predation. This is the figure where I got that figure right here. I mean, this is a laboratory experiment where we directly observed a unicellular organism becoming an obligate multicellular organism. And this involved a number of unique new traits. This group of organisms, it's a type of green algae called Chlamydomonas, never had this thing called an extracellular matrix. An extracellular matrix isn't even something that all multicellularity things have. Animals have it, but other multicellular cellular things don't. This is a completely new example of an extracellular matrix. Reproduction via multicellular structures called propagules. That evolved completely new in this species. These new traits involve new genes. You have to have novel genetic information in order to get this new stuff. And we could go top to bottom looking at things like that. Let me end this screen share here. I have another example here of a plasmid. Kent, do you know what a plasmid is? Everyone in the audience know what a plasmid is? Everyone familiar with that term?
It's been years ago that I studied that. I don't remember the details now, but anyway.
I think it's this one right here.
One topic at a time, though, Dan. Can you go back to your slide of your algae? Is this a colony of algae working together, or is this actually one single organism?
It is one single organism, and we know that that's the case because in a colony, what you need to have is basically there's kind of competition within the colony because each thing could live on its own, and if you don't reproduce, you kind of lose out. But in multicellular, you have an agreement between all the cells in the organism that, like, look, we're all genetically identical, so you over there, the germline, you're going to reproduce, and that's okay with me, the skin cells, because you have all the same genes anyway. And in this example with Chlamydomonas, they reproduce as multicellular units, but not every cell in that group reproduces. So they have specific cells that are responsible for reproduction. It's not just a colony, it is true multicellularity.
The rest of the debate kind of follows this same format. Kent will deny that multicellularity can arise from a single-celled organism, and then Dr. Dan will show evidence from this paper and rinse and repeat. The interesting thing is that Kent has already seen this paper in a debate before with Erica. So Kent was pretty annoying as usual, but I'd like to give a lot of kudos to Dr. Dan for his performance in this debate. He really whooped Kent. One thing that was pretty irritating, though, was being in the live chat and seeing Standing for Truth and the other mods calling Dr. Dan a liar and other names during the debate. And then during the Q&A section, they basically asked Dan like 9 questions and asked Kent 1. I was tagging SFT in the chat with my question over and over again, but it never got read because during the Q&A they were reading super chats from the mods that were just a bunch of, gibberish memeing back and forth. They weren't even questions. I asked Standing for Truth about this, and he told me to quit complaining. I guess he's not very good at taking constructive criticism. All in all, it was one of the best debates I've seen in a long time. Unfortunately, Kent brought out his old excuse of, I'm not asking for my theory to be taught in public schools. And Dan didn't really do a great job at pouncing on this point like I would like to, but that's something that pretty much everyone does against Kent. It's his ace in the hole. It's his, last resort argument that he'll make, and he brought it up a lot during this debate, which I think just shows how badly he lost. Now, what I want to know is, when is the Conspiracy Cats rematch? Kent has been putting this off for a long time, and I'm interested to see what channel this debate would be on, because I know Standing for Truth has been helping Kent with ERVs, so hopefully it won't be on his channel, but I guess we'll see. Anyway, I'm going to end the video here, so smash that like button, subscribe to the channel, ring the notification bell, check out my Twitter and Patreon, and have a nice day. Also, don't forget to pay your taxes.